opens up inside the polymerase complex. One strand goes up here, and this is not going to be used to synthesize the mRNA. This is the template strand. This one is going to be used to synthesize the mRNA. And it passes by the active site, or the catalytic site, where the actual synthesis is going to happen. And the heart of that is this magnesium ion that held in the center of the complex. Okay, so passing by the magnesium ion, nucleotide triphosphates are going to come in through this funnel and core complex. So remember in the movie, those yellow single nucleotides flying in that look like they were getting sucked up on the back of the they go up in here. That's where the ignition happens. That's where the magic occurs. And they get added to the base pair with the template strand, and you start to make the new mRNA. As this gets longer, it comes out the top here. So that's where the messenger RNA exits. The new nucleotides are being added here. So which end of the molecule is it? 5' or 3'? So 3' this is the 5' end. This is the template that's moving by, and it goes out, and it joins outside 3 amines. So it reestablishes double helical structure and goes off and down. Yeah. Yeah, that's a passive process. Yeah.
Alright, so DNA, like, little transcription thingy goes from 5' to 3'.
So as it moves along the DNA molecule, it's going 5' to 3' with regard to one strand, but 3' to 5' with regard to the other strand. So we always have to specify which DNA strand we're talking about. The RNA molecule is 5' to 3', so the template strand is going to be the 3' to 5' to 3'. The opposite. I'll show you that. Okay, so hold that question. I'll show you another slide that clarifies all that. Okay. But that's a good question. Yeah. Correct. The RNA is going to stay single stranded and come out here. These two DNA strands, the template strand and the coding strand, are going to radial up here and exit. So remember, this whole thing's moving along the DNA like this, and stuff squirting out the back of the goes. So, as in many aspects of this class, we want you to think about, we show you these static pictures, and sometimes we can show you movies, but we want you to think about these as dynamic processes. And hopefully in the movie you get the sense that this happens super fast. It's a highly dynamic process. Yeah. Okay, so the question is, are there always a lot of nucleotide triphosphates disporting around the cell? The answer is there needs to be quite a lot, or you can't make messenger RNA. Now, there is in some cases, a cell that's under stress or starving will regulate transcription by limiting the pool of nucleotide phosphates. By and large, there's always a lot of them. Okay. So that's what the polymerase complex looks like. In your cells, there are 3 different complexes, but in bacteria, new bacteria and archaea, there's only one. The reason is that in those groups, there's a single complex that makes all the RNAs. But in eukaryotes, that would include the yeast you're studying in class and yourself, there are 3 different enzyme complexes, but we're mostly going to be concerned with the middle one, RNA polymerase 2, which makes the messenger RNAs. RNA polymerase 1 makes the large ribosomal RNAs. RNA polymerase 3 makes transfer RNAs, the smallest ribosomal RNAs, and a bunch of other small RNAs, including, to the best of my knowledge, most of the microRNAs. That's still current true. But also some other non-coding RNAs that we don't know as well about, but this is a major division of brain. In a living cell, as Steve pointed out, most of the RNA is actually tRNA, ribosomal RNA. Messenger RNA, it depends on the cell type, it depends on what that cell's doing. It's going to be somewhere between 3 and 5%, 5 is a good middle number, is going to be messenger RNA. So this isn't most of the RNA that's produced, but it's for most of the regulation. And we'll talk more about the regulation component later, but we're really interested in polymerase 2 because its activity, and the regulation of its activity, is going to be the difference between a muscle cell and a nerve cell, or a cell that's responding to stress and a cell that's doing its normal thing, or a dividing cell and a non-dividing. All of those processes involve regulation of transcription, and the particular kind of transcription that will be regulated is the production of specific messenger RNAs, and they are produced by polymerase 2. So RNA polymerase 2 complexes is what we mostly care about for regulation of transcription. If you look at the structure of these polymerases in the 3 different domains of life, they look pretty similar. I don't want you to remember or memorize the subunit structures here, but what I want you to get is an appreciation for the fact that they're structurally very similar. All of them have a beta and beta prime, or depends on the nomenclature, alpha, beta DNA, and 1 and 2, it's the same thing. They carry out the same functions, they have roughly the same shape, and then there are some additional proteins that are stuck on there. Notice something interesting though. Archaea and eukaryotic transcription complexes look much more similar to each other than eubacteria and archaea, even though these are what we call these prokaryotes. They're single cell, don't have a nucleus. In terms of cell structure, bacteria, eubacteria, and archaea look very similar. But in terms of transcription particularly, archaea are much more like eukaryotes. And this is something we'll see later when we talk about other aspects of function in archaea. But in terms of transcription, they're much more like eukaryotes. The only other thing I want you to take away from this is that the origin of this complex, the evolutionary origin of this complex, is shared by all of these groups. And since it's something else, the commonality in all life is that back machinery is essentially modified slightly for greater or less than years. So they share a fundamental set of properties. There are some differences, and I'll point out some differences. Mostly what we're going to be talking about today is from eubacteria. Steve's going to talk about eukaryotic regulation for the end of this set of lectures, but for the first couple of days, we'll mostly be talking about eubacteria. I'll try and tell you when they're different, but that's by and large what we're going to be talking about. Okay. Stuff to know about transcription. As I mentioned earlier, you have to melt the DNA or open it up in a local region for the polymerase to gain access and start adding those ribonucleic. You should now understand that the process of addition happens sequentially. One goes on, and then another one, and then another one, and another one. The energy to do that comes from the nucleotide triphosphate itself. So you're converting a triphosphate to a monophosphate. That's an energetically favorable reaction. The direction of extension is from the 5' end to the 3' end, and you're adding covalent bonds so they put together. Phosphodiester bonds. The decision about which nucleotide to add is based on hydrogen bonding, the base pairing with the template strand. So if the template strand has an A there, then you can bring in a U. If it's a C, you're going to bring in a G. So make a new bond, and then go to the next one. At the end of the day, that RNA DNA duplex has to melt to release the mRNA so it can go off and become transcription. Yeah. If there are, there's a pool of 4 ribonucleotides in the cell and a separate pool of deoxyribonucleic acid itself. And one of the functions of this enzyme is to control the different. So if it's that's a good question. If it's an A on the template, so it's not going to bring you a T because that would be a deoxyribonucleic acid. Did everybody catch that distinction? This is something I should have said, but thank you for bringing this up. This enzyme distinguishes between the pool of deoxyribonucleic acids, which you're going to use for DNA replication, from ribonucleic acids, which you're going to use for RNA. Let me tell you here. During the attack, we're going to talk about the initiation, how it happens. So, particularly eukaryotic cells, you know this pretty well. There's a special protein that actually binds and kinks the DNA to pop it open, and that helps with all right. So these are the three major steps, as I mentioned earlier. There's an initiation phase, involves getting the polymerase onto the DNA, and it stutters a little bit at the beginning, and I'll talk about this in more detail in just a second. It gets on, it kind of jumps around a little bit, and it doesn't really get started for a while. And then once it gets about 10 bases in, then it really goes. We call that next phase elongation. That's where it just cranks out most of the messenger RNA. And then at specific places, it receives a termination signal and falls off. So those are the three steps. And I'm going to go through each of them in detail. So hold your question on them now. That's where we're going. And here's the same thing in pictures. Okay? So nothing different, just to give you a visual sense of what's going to happen here. Here's a region of DNA. The polymerase is going to attach its specific site. It's going to melt the DNA. This is a current process really. You're going to make a couple little pieces of DNA which are stuttering, not doing a very good job. And then finally you get about 10 bases. That's called clearance. And now the complex can move across. That's elongation. And then it reaches the termination signal and falls off. Yeah. Okay, so it's a great question. How does the polymerase know which strand? The answer is that binding happens at very specific places. So it's going to attach at these sequences called promoter sequences, and that's going to, the DNA is going to open up, and then it's going to move in a 5' to 3' direction. So to move in that direction means that the template strand, it has a flare, it's facing its own direction. That becomes the template strand. So one of the functions of a promoter is to position the attachment correctly and orient it correctly. It could face that way or that way. If it faced that way, it would make one necessarily. If it faced that way, it would move in a 5' direction. So there's an inherent polarity to it. 3 positive templates. 3 positive templates. So there are specific sequences that dictate where transcription starts and where it ends. The place it starts is called the promoter. This is where polymerase binds. Yeah. Yes. And our definition, the broad sense definition that we're using here is we're saying regulatory sites, whether they are promoted or not. Broad speaking, so the polymerase will bind here, will start moving across this region, and will fall off at the terminator site. So there are specific places where transcription starts, and there are specific places where we'll talk about what the transcriptions are a little later, but just be familiar with the terminology and the fact that the extent, the position, and the extent of the messenger RNA molecule are determined by And of course, the direction of transcription is you start here and you move across, or you reach a terminal. This is the same image, just shown in a different way with the polymerase on the DNA doing its thing. So here, this in gray is the double-stranded DNA. There's the open duct part. It's diagrammatic. It actually happens inside of the format. And then that's the messenger RNA molecule coming out. So the polymerase is moving along the template strand, producing the RNA. It's moving in a 3' to 5' direction on the template strand. The messenger RNA in a 5' to 3' distance. So the growing chain of RNA is happening 3' there. This is the first nucleotide, second, third, fourth, fifth, the most recent one. And the next one will be added here. If the DNA, to get back to the question that was asked earlier, if the polymerase were facing the other way, it would make a different molecule. So one of the functions of the promoter is to orient the polymerase to face the right way. So not only is it positional, it's a polarity. Yeah. The synthesis of the strand is 5' to 3', but the movement along the template strand is 3' to 5'. They're in opposite direction. Talk about the RNA being made 5' to 3'. The movement on the template is 3' to 5'. Yeah. The new nucleotides, that's a 3' end. So you're adding at 3' end, so the movement is 5 to 3. It's going, the 5' the oldest part, the newest part's the 3, so you keep adding 3'. So it's growing in that direction. So the template strand is where the base pairing is happening. The coding strand is not part of the process. It's going through the polymerase, but it's not being used as information. Question? If the promoter were oriented in the opposite direction, it would go the other way. It's with respect to this gene. So that's a really good point. So this is something to keep in mind. On a chromosome, that piece of DNA could be, pretty big. Some of your chromosomes are that big. Okay? Some genes face this way, and some genes face that way. Coding and template are with respect to the gene you're talking about, and not locally across the whole chromosome. So genes face both directions. That's why it's important for the promoter to not only position the polymerase when it attaches, but orient it in the correct direction. So the question is, could two genes be essentially in the same place but facing the opposite directions? And the answer is that does happen sometimes, particularly in bacteria. But even in your genome, there are some overlapping genes. So the same nucleotide read in one direction is part of one gene, and read in the other direction is part of another gene. There are a few of those. There are not many in the human genome, but there are a fair number in a lot of bacteria. Viruses live in small time. They're very compact genome. They see a lot of information in small time. Okay, so template and coding. Now, sometimes people find this terminology a little bit confusing. The most important thing is to remember template is literally that. It is the template used to make the mRNA molecule. This is what the RNA polymerase reads to get the base pairing right to make the mRNA molecule. Coding has the same sequence as the mRNA was used. It's not used in the process, but it has the same sequence because of the complementarity. Okay? So this has the same. When people write the sequence of a gene, you're looking at the DNA, they say, this is the sequence of this gene. They don't usually only giving you one strand, they're giving you the coding strand. They're telling you what the mRNA is going to look like if you make from that gene. But it's not template, it's like the opposite strand. get those things straight here. That's a little confusing. Make sure you understand the difference between template and coding, and make sure you understand 5 primitive the orientation of the process. Okay, so this is a pretty important set of orientations to get straight to. Okay, any questions about what's going on? Yeah.
So basically with the two rows of DNA or whatever, you're using the template strand, which is the one on top, to get the one on the bottom. So you're replacing the T with the U.
Correct. The template strand is going to the main. You're going to see similarities between this process and DNA replication, which Steve will talk about a little bit later. So you're going to get a photo here. If you understand this, you're a long way toward understanding DNA replication. The same basic set of processes is going to happen using a template to make a copy. Yeah. Because the other gene, you had a so there's a promoter over here where that polymerase is bound and sort of moving this way. You could have a promoter here, and it would go like that. And in which case, we would have to reverse these two conversions. Okay, so now let's talk about initiation. So I've already told you the first point, so you should already get this. Transcription begins at specific places. It's not random where the polymerase RNA polymerase actions. It's only going to time to certain spots. Those spots are determined by the local DNA sequence. Okay, so that binding is going to happen based on the local sequence of the DNA. RNA polymerase is not going to bind to those by itself. Even though it uses those as cues, it needs helper molecules to get on there. Helper proteins, not just going to attack the foundation on there. The reason for that is the process needs to be controlled. We'll talk about that control later, but just be aware that RNA polymerase doesn't just attach to those sites on its own. That regulation happens through a set of proteins called transcription factors. In new bacteria, we call those signal factors. I'll get to that in a second. Just be aware that transcription factors is a general term for a protein that helps regulate its transcription. And they also bind at specific sequences. I'll tell you a little bit about that, but we'll get to that in more detail. Just be aware that that's what's going on. And that transcription factors not only bind to DNA sequences, but they interact directly or often indirectly as well. Help get the polymerase onto the DNA. So they guide it onto the DNA and help it attack. And you can't do that very well on its own. A lot of this is where the regulation happens. Again, that's something we're going to talk about Wednesday and next week. Just be aware that that's part of the initiation process. Promoter sequences I told you about earlier, those are specific sites where the attachment happens. And this slide illustrates some of the features of promoter sequences in new bacteria. What you have here are the DNA sequences for a couple different genes. So that's one gene, 2 genes, 3, 4, 5, 6, 7. And what you can see is that if you line them up, where you have the start of transcription at a particular nucleotide, there are some similarities in those sequences at specific places. There aren't any similarities here, or here, or here beyond the random. But at 2 places, there are a greater than random degree of similarity. Not identity, but a greater than random degree of similarity. You'll notice this when you're first starting sequencing DNA around bacterial genes. And that led to the recognition that these were, in some senses, functionally important sites for regulating transcription. So we call these the -10 sequence and the -35 sequence. They're not always in that exact location, but they're usually near there. And out of this, we have a consensus. The consensus is not an exact match, but it's close. So the sometimes there's a difference. There's a C here, but normally it's a T. That's the same thing over here. It's usually a T, but sometimes a C. That's called a consensus. So one of the important things to understand is that transcription factors don't bind necessarily to the exact things. They bind to consensus. So sometimes people have a little bit of trouble thinking about what this consensus means. It just means that transcription factor is picky, but not super picky. These are the sites that the sigma factor of the transcription factor is recognized. And you'll notice these are not the same sequence, and they're not palindromes of each other. If this over here were the opposite of that, you would be able to orient properly. But because they're different, they have an inherent polarity. They're going to face the transcription factor in that direction. They're different from each other. So they provide the orientation. So not only are they positioning the transcription factor, the polymerase complex, upstream from near the beginning of the gene, but they're making sure it faces in the correct direction. So instead of rushing away from the gene, it runs into the gene. That's why they have to be different from each other. Polarity. I'm going to say more about how that works later. I just want you to get a sense for what the promoter sequences look like in a eubacteria stuff. So this is what happens. There's the DNA polymerase. Remember, it looks a little bit like that. Here's a sigma factor that's going to help it bind. And the sigma factor contacts at the -35 to the -10. So you see it's touching the DNA in two different places. And that's helping the polymerase get onto the DNA and face the correct direction. It's over here. So when it starts going, it now knows when you the template strand and things move in a 3 times, 5 times direction on the template strand. It can only move 3 times or 5 times, but it's not going to get on the wrong strand. It has to get on, there's only one strand for it to use. That's going to be the template strand. It only moves 3 times, 5 times along the template strand. The other strand is getting wrong direction. It's like a racket. It can't go against the racket. So far? Okay. Let's stop for a second and just think about why secret factors are used in the process of transcription information. I'll just give you a clue. There's more than one right answer. All right. Let's do think, pair, share.
Raise your hand. Don't know anything about sequencing. Did you hear that? Yeah, right. So the answer was A, it helps the polymerase along the DNA once it's arrived, and the second is it helps the polymerase actually find the promoter genes.
Good. Okay. Anything else?
Who else?
Okay, so that's also true. So the sigma factors are not only helping to attach the DNA polymerase to the DNA, so RNA polymerase to the DNA, but also exerting some regulation. So that's a very important function. Both of those are important functions. We'll talk more about that, the regulatory aspect. Anything else? What's the other thing that you should now know that sigma factors have to do? Right. Polarity. That's the other thing. Okay, so it's helping the RNA polymerase to bind to DNA. Remember, it can't do that very well on its own. It's helping regulate which genes are being transcribed, and it's helping the RNA polymerase to face in the right direction, right, for polarity. Those are all important. That's good. Oh, yeah. Right. So the question is, could RNA polymerase bind without the sigma factor and start transcription? The answer is that at a very low rate, there is that kind of phenomenon going on. So even though the sigma factors are, their function is to help it bind and to orient the proteins, a low amount of what we call leaky transcription, molecules per cell kind of thing going on all the time. So this is a facilitator, not an absolute requirement. But it's a huge difference in how much mRNA is made in sigma factor there. Okay, so here's just a picture, and this is the last section to talk directly about this process. We really want to talk about the production of the mRNA molecule. But for the sigma factor, for the polymerase to get on, the polymerase is this gray stuff up here, and the sigma factor is the blue. You can see part of the sigma factor is contacting the -35, and part is contacting -10. So obviously, if you had the opposite order, they would attach in the other direction, and the other place goes on. So the contacts at two different places. The hydrogen bonds, the base pairs is reaching into the major groove. Again, we're going to talk more about this on Wednesday. Reaching into the major groove. You're having hydrogen bonds attached. They set up the specific side chains on an alpha-glial and particular base pair, just like the transcription enzyme we talked about earlier. Same basic concept. The recognition of hydrogen bonds and stabilizing the interaction. Instead of having the cleavage of the DNA happen next, as you would with the transcription enzyme, then you've already essentially accomplished the goal. Get the RNA onto the DNA that opens up and starts. Yeah. Polarity is so DNA and RNA are polymers, and the ends are different. And so we use the polarity to say we have to interact with them. of the two, whenever there's a DNA-DNA hybrid or an RNA-RNA hybrid or an RNA-DNA hybrid, any of those go in the opposite direction. So one strand is going 5' to 3', the other one's going 3' to 5. That's all of them. They're always opposite. Anti-parallel. No, but it's always out of the that I don't think. Yeah. It's not forming hydrogen bonds. It's not the same hydrogen bonds that are base pairing. There are other groups on there that it's expected. In other words, it's not disrupting the base pairing to do this. Is that your question? Yeah, it's not disrupting the base pairing to do this. Okay. So now we're going to assume the polymerase is attached to the DNA. It's already on there. Okay. What happens next? First of all, the DNA polymerase complex itself changes conformation after tightly and it melts the DNA. It's all happening more or less simultaneously. And then they begin to synthesize RNA. And they will synthesize short stretches of RNA. I'll show you this in pictures in just a second. The uRNA 3, 6, 8, when it can get, and those will fall off and it'll start again, and it'll start again, and it'll start again. And eventually we get up to about 10 or 11, then it goes, and then we're talking about elongation. Just talking about that initiation part, what's going on there. Once it clears that 10 or 11 base pair, the sigma factor is going to fall off in elongation. Let's look at those little steps. So here's again, just picture with what I said. The polymerase is coming on, there's the sigma factor helping it attach. It opens up the DNA and it starts to make short RNA molecules, and those are going to fall off. It's called a Gordon initiation. Whoa. Gordon initiation. So these small fragments you can actually detect each of the cells and make a box of information about how that's done. We're not going to ask you to understand that in detail, but if you're curious, you can see how people learn about this. But the point is, it's stuttering. It's not really getting started very well. It's not well understood why this happens, but it's probably part of regulating the initiation. So potentially another kind of regulation. Once it clears about 10 bases, the sigma factor falls off and it starts to explode. Yeah. I would read this too literally. Yeah. Yes, it's backing up and starting over and over again. So the start site, the place where the first nucleotide of RNA is added is the same nucleotide concentration. Now, I will just say that in bacterial genes, initiation almost always starts in exactly the same place. Your genome, things are a little messier. Some genes actually have 2 or 3 different start sites, but physically different promoters, even though it's the same gene. And there's sometimes a little bit of stuttering to where it's starts. Like the first approximation is always the same person. And there's a molecular assay you can use called nuclear lemon assay to measure that. Yeah. Okay, so the question is that first part genetically useful? Is it coding for proteins? And the answer is it is not. It's what's called an untranscribed region. 5 times untranscribed region in UTR. We'll get to that later in that. But it's anywhere from a few hundred bases to but again, we're getting a little ahead of ourselves. So this is what that stuttering is going to look like. The polymerase is on the DNA is melted, and it's starting to add a few nucleotides. The sigma factor is still attached. And it makes it a little bit longer, until finally it gets long enough that it can come out the back end of the molecule. And that's when you lose the sigma factor, and now it's ready to really go. Okay, so that's the abortive initiation phase. So now we're done with initiation. So the key things to think about in initiation is getting the RNA polymerase complex onto the DNA, positioning it correctly, orienting it correctly, and getting the first 10 bases synthesized. At that point, the sigma factor falls off and it's ready to go. Now I'm bringing it along here. So any questions about that initiation process? Yeah. These steps are happening with slightly different variations in areas as well. So there's stubborn in your cells, there's stubborn in these cells, there's stubborn in corn. That the stuttering process, the necessity of attaching in the sequence-specific manner, which is generally shared. Sigma factors are a specific feature of the way you bacteria do it. In your cells, there are other factors that guide it on, and generally with this. Okay, now I'm going to show you. Oh, sorry. Question. It's just that it makes a couple of nucleotides and doesn't work, so it goes back to tries again and tries again. Eventually, it makes enough that it can clear the early region and so forth. I'm just going to show you the same thing with other diagrams just to reinforce it. There's nothing new here. It's just looking at it again and thinking about what's going on. So here's the sigma factor up here in orange. Here's the DNA helix. This is the polymerase complex. It starts to open up the DNA, to melt it, loops it around the rudder. There's the rudder. This is the cleft, the jaw behind it. It's now opened up. There's the magnesium ion. There's the first little piece of RNA being synthesized. So we're starting abortive initiation. Make it a little bit longer, and eventually, once it gets past about this point, that's 10 bases. And now it's out the back and it's ready to go. This flag opens up and the sigma factor is going to fall off and it's going to be able to carry out. Maybe I didn't tell you before, this is different things to think about it. Go back and look at the notes and sort of get it all straight in your head. And kind of relate this back to the anatomy of the polymerase complex I talked about earlier. Yeah. The cleft, the jaw is behind, so the DNA is coming into the cleft. That's the rudder. So when the melting happens, the DNA goes around both sides. The template strand is going to go here, and the coding strand is going to go there. That's the active site. And then this is the DNA is going to exit there, and the RNA is going to exit there. This is the funnel, and the nucleotides are going to come in. There's the pore. So the funnel is, it gathers up the nucleotides and then brings them up here to the active site through the pore and then the active RNA. Yeah. Up here. Yeah. The DNA is closed, the RNA polymerase is closed, nothing's going on yet. There's no RNA being synthesized. Starting to melt. Melted, you're starting to add RNAs, that's open. And now the elongation complex is the next clear avoid. So closed, open, and elongation. And I don't really, I don't really care so much if you understand the terms and you understand the process here. Yeah. All right, the question is, the sigma factor is also responsible for melting, but that's to my knowledge they're not. Now, in eukaryotes, there is something like a sigma factor called non-binding protein, and it absolutely is responsible for the initial melting. So that's a difference between the two. I don't want to do that. The whole complex does that. essentially undergoes a conformational change, extends the DNA 60 degrees, so it goes like that. What that does is, just like the restriction enzyme, that thinking of the DNA helps to open up the double-stranded helix, and begins, that's kind of the where the thin edge of the wedge comes in and starts to unzip. So that's one way that DNA is physically bend it. It pull apart the double strand, I mean the hydrogen bond of the double strand. Okay, so we're ready to talk about elongation. Yeah. Conformation change. As the RNA gets long enough, and this is not very well understood, there's a piece of the sigma factor blocking it during the order of initiation. This is something you do not have to know. This is to answer your question. And there's a piece of the RNA polymerase complex that blocks it. For reasons that we don't understand, those move out of the way, and the growing RNA changes past them. That triggers the sigma factor. It does in the sense that it helps increase the frequency with which it is binding to the DNA. So it's helping with an absolutely essential step. But following binding, I don't want to do that. Okay, elongation. This is a super fast process. It's a 50 nucleotides a second in bacteria, 30 nucleotides a second in your cell. That's 400 RPM in a bacterium. It's like 300 RPM in a bacterium. That's basically how fast your engine's running when you're on idle in the car. It has to be accurate. And it does this too. It can't mess up. It's putting the nucleotides in. We'll talk about a little bit about how that happens. Has to be a super fast, super efficient process. However, it's not completely smooth. And I'll show you in a couple slides physical evidence for that. It's not as if it always is going at 50 or 30 meters per second. It has a pause, and then it starts up again. There also does proofreading. While it's doing this, if it detects a misincorporated base, accidentally adds the wrong one, it will stop, back up, replace it, and go on. So it's not an absolutely smooth process. As it's going through the DNA, unwinding it, it's going to have to be opening it up in front and closing it behind. That introduces the problems that Steve alluded to last week, which is our cognitive autopsy, briefly about that. And that there's specific enzymes that are dedicated to making sure that happens. These are some of the things to think about during elongation. So here's what the elongation complex looks like. You should now be pretty familiar with this molecule. There's the entering DNA, so the whole complex is moving in to the right, okay? It's moving in this direction. DNA is coming in, or if you think of this as fixed, the DNA is coming in from that side and going out that side. There's the new messenger RNA molecule, there's the catalytic site, there's the rudder, this is the healthy DNA, okay? That's the elongation complex. This is, unfortunately, this diagram is flipped around, but I'll flip this around and tell backward. We're now moving backward. I apologize, it's the only thing I'm showing. I think I'm going the wrong way. But I want you to get the sense of what's going on in the transcription bubble. Okay? So here's the entering DNA. It melts. There's the rudder would be right there. Okay? This bubble, the so-called transcription bubble, is a single-stranded part. It's about 9 or 10 bases. And in that bubble is an RNA DNA hybrid. So this is this brown is the RNA molecule that sits inside that green here. Again, I'll put this around so it's the same orientation to help you see this. And this is the most recently added nucleotide. This is the first nucleotide. So it's been growing in this direction. The DNA exits the polymerase complex and bringing the others back to the double stranded DNA. continuously moving along the DNA, opening up the base of the time and closing the base of the time behind it. So, okay, so I lied. It's about 14 bases on 14. A hybrid's about the opposite. A hybrid's about 910 bases long. And then you have reformation of DNA hybrid behind the bubble. Okay? Yeah. Right. So this is just don't take these diagrams too literally. What we're trying to get here, so you're right. I mean, it's inside right there. You mean it's on the inside of that loop. I think this is just drawing this way to make it clear. So I don't care that you understand the three-dimensional orientation of these molecules. Oh, these little numbers, ignore these little numbers, yes. Ignore these little tiny numbers. The 5 prime to 3 prime is correct, but don't worry about these numbers. We're not going to have to do that. But there's a different number system normally this would be base pair number 1, 2, 3, 4. That's a transcription bubble. Remember, it's constantly moving. And inside the bubble, you have an RNA DNA hybrid. And then as the bubble moves, behind it, you're really looking to make a DNA hybrid. Back to the normal double-stranded DNA behind the bubble. All right. So I want to show you how we know it's pausing. We only have a couple minutes, so I'm just going to run it. But essentially, this is using a physical method where you image 2 beads, and between them, they're passed by a single molecule of DNA anchored on one bead, and there's a polymerase on the other bead, and the polymerase to DNA, RNA polymerase to DNA, interacts with each other. That's the only way you see these coupled together. And we can actually measure the distance between them. Okay, so the first one is the video. There's one bead, and there's the you can't see the DNA molecule between them, but it is strong. It's physically attached to one and only interacts with the other. And when transcription happens, it actually pulls the other bead toward it, because the polymerase is moving along the DNA. And if you measure that in time, it's a pretty continuous process. Okay? So this is time, this is distance. It's a pretty continuous process. However, sometimes you need to get situations where it's hot. It's not like the video is hot, the movement is hot. Time's actually absent there. And now it goes on. So you have this period in here where there's no movement, even though time is passing. Okay? So that's just showing you pausing in experimental situations to show you how that happens. Alright. I'm going to take just a second to show you what the issue is with pausing. Alright, so as Steve mentioned earlier, I have you read a polymerase molecule. Okay, so if you could grab maybe a marker or something like that. Oh, perfect. Okay. So here's DNA. DNA is twisted, and if I keep twisting this, it's going to supercoil. Just a second. So supercoil, you can see supercoil starting to develop now. Okay, these are supercoils. They're doing that. That's a supercoil. And that's all it is. If I keep twisting, I'm going to get more of those. The more I twist, the more supercoil I'm going to get, right? So I keep twisting, it's going to start doing that all over the place, right? Now, open up the, so now here's Mr. Polymerase is coming in. And he's going to open up the DNA strand, which you can barely do, right? Now, if he moves toward me, what's going to happen? It's going to get relaxed behind him. And see, he needs help. See, look at all the supercoils here. It's getting more supercoils here. It's more relaxed. Positive. Okay? All right, thank you, sir. All right, that's all we're talking about here. Okay? So trying to do this, do that as a demonstration, so I want to skip through the last couple items here. Okay, so supercoiling happens in front of the polymerase molecule in between. This is just a picture to show you what the physical demonstration was that Stephen I just did. As the polymerase moves, it's going to heat up the DNA in front of it. So you're getting positive supercoils in the direction it's moving, and you're going to get relaxed or negative supercoils. So just think of, just remember that kind of as a mental picture, why the cell needs to look at some of the processes to take care of that thinking. Otherwise, it's going to bump into stuff. That's part of the reason for pausing. Another reason for pausing is the DNA, as it opens up, sometimes makes little proteins, all kinds of reasons. And when the protein is relaxed, it can stop the protein so that the track moves in the So those are the reasons for pausing. As the bubble moves, here's your bubble, there's your RNA DNA hybrid. The positive supercoils in front are resolved by an enzyme called DNA gyrase. It basically cuts one strand, flips it over to the other side, reattaches it, so it's with the leaving the supercoils. And then behind, a different enzyme called topoisomerase is adding, is again cutting and reannealing to reestablish the connected positive supercoils. So these are just some pictures of those enzymes. Here's topoisomerase 1. There's the DNA double helix, and there's the molecule. I don't really care that you know a lot about Connie's work, but there's a lot of things that to study exactly how these molecules do the relieving and the addition of extra-nuclear. Last thing about elongation is the proofreading that happens. As the polymerase moves along DNA, it can detect if it made a mistake, because there's, remember, in the transcription bubble, there's a place about 9 or 10 bases where you have a RNA DNA hybrid. If you misincorporate a base, what do you think is going to happen to that RNA DNA hybrid? Yeah. Won't be quite as tightly bonded. There'll be a little bubble in the middle where the two bases aren't correctly joined. The polymerase complex can detect that. It moves back, it backs up. Well, I guess it's not that big, so it backs up. So what happens is that the molecule that was, this is where addition's happening, it backs up, it actually pulls this back in to get to the base that was misincorporated, cuts it off, and it does it again. So that's how the proofreading happens. It's not a super efficient proofreading process, but it's pretty good. This is to resolve the supercoiling we showed you earlier. Here's the polymerase complex moving along the DNA. There's, sorry, this is to get rid of the chromatin that Steve talked about running. There's DNA bound around nucleosomes. Remember, when transcription is happening, DNA is in the 30 nanometer fiber. Sorry, it's in the beads on the string. It's not in the fiber. It's in the beads on the string configuration, but the bead is still there. It's not in the beads on the string. And to do that, there are proteins called chaperones that pull the nucleosomes apart, disassemble them, and then reassemble them back on the other side. So not only do you have enzymes dealing with the positive and negative supercoiling as it moves through, you've got proofreading going on, you've got nucleotides flying in and out, and you're disassembling content in front and reassembling it behind you. It's an incredibly complicated process. The video you saw at the beginning didn't talk about pausing, it didn't talk about proofreading, it didn't talk about chromatin. So it's even more complicated than it looks like when you first saw it. Alright. We can do termination in about 3 minutes because that's all the time we have, and there's really not a lot to do. Transcription starts at specific spots and also ends at specific spots. I'm going to tell you about 2 different mechanisms that operate in new bacteria. The truth is we don't know very well how it happens in eukaryotes, like yourself. There are some ideas and there has been some work done, but we don't understand as much about the process. I'm just going to tell you about the transcription termination mechanisms that we know how to do that. We understand that. And I'm just going to skip over this in pair share. So we can think about. I've put in some answers here. So you can just jot these down. But basically, you can imagine that you don't want transcription to go on forever because you're building a long molecule with energy. Remember, this big phosphate is being added every day. You had to put energy into building this big phosphate. So there's an energetic argument. If you go too far, you actually get into the next gene. And in eubacteria, translation can start in the middle of the transcript. So you could actually turn over one gene by mistake if you didn't stop the process. And in eukaryotes, you could end up having some problems with mRNAs, so there's a bunch of reasons why you want to terminate it. But basically, two pretty well-studied mechanisms in eubacteria. One relies on making hairpin structures out of the RNA molecule itself. That's called intrinsic termination. It's called intrinsic. It's all the stuff you do in the RNA molecule. And the other is called extrinsic termination. You use protein complex called drug. In both cases, what's happening is the polymerase is going to bump up against a physical barrier and fall off. That's really all you need to know. There's just two different ways of having that happen. You either bump up against the hairpin or it can bump up against the growth protein. In either case, it's going to fall off. So here's what intrinsic looks like. So here's the RNA, here's the transcription bubble, there's the RNA DNA duplex. As it goes along, it forms a bubble, and that sticks onto one particular subunit of the polymerase and grabs it and sticks with it. Okay, so that's how it happens with intrinsic termination. Now with extrinsic, as the RNA is being produced, it attaches to this complex, it feeds up to the central hole and binds to these little subunits. And then that loops around and attaches onto the polymerase. Same thing. They just essentially physically stop the engagement. Those are the two mechanisms. This is just a picture showing you the structure of growth factor and the U chain coming up through the middle and getting tangled up on top of it. All right. I'll see you on Wednesday.
Was your mom's?
Yo, are you going to the...